Well, thank you everyone for joining us. This is now the fifth session of the autumn lecture series, and we've been looking at quantum mitochondria. Now, today's session will consider mitochondria and fields, the implication for space exploration. Really a fascinating topic, and especially with the launch of Artemis 1. Was it this morning or last night? And we're delighted to welcome Afshin Beheshti as our speaker today, and Alistair Nunn and Steve Thorne have prepared some slides, but I believe Alistair is going to make the introduction to Afshin today. So our introductory deck was prepared today by Alistair and Steve. Alistair Nunn is our Director of Science for the Guy Foundation and visiting professor at the Research Centre for Optimal Health at the University of Westminster in London. And Steve Thorne is founder of the Copernican Project, a non-profit organisation centred in Berkeley, California, which is investigating a possible coupling between metabolic processes and strain effects generated by periodicity in the gravitational potential due to the Earth's orbital motion. So I think without further ado, I'll hand over to Alistair to share his screen to introduce our speaker for today. Alistair.
As Jeffrey's introduced Afshin, he's the bioinformatician and principal investigator at KBR NASA Ames Research Centre, and one of his interests is in studying the role of circulating mRNA and mitochondrial disease. And really this ticks many boxes with us because we've really come from the mitochondrial direction too. But he's also looking into COVID-19, which we've been dabbling in a bit too, clearly space research and also cancer. So these are a lot of crossover points, and I think some of these are quite an interesting crossover point, as I'll try to talk about. So why are we so interested in this? Well, we're beginning to think that maybe, and this again, we said this before, but one of the missing components in homeostasis in biology, we know about chemical homeostasis and pharmacology, but maybe non-chemical homeostasis and the role of fields. And I recommend, if people haven't read this, read this book by Fels, Siffer and Schockerman, which talks about the fields. It's a great introduction to the idea of fields in general in life and cells. So sorry for the slightly strange diagram, but we're certainly going to be discussing today again the role of mitochondria. And one of the things that is becoming very apparent is that mitochondria clearly do a great deal more than simply make energy or capture energy, ranging from, and they're involved in inflammation, stress, epigenetics, management of ions. It's central to immunity, obviously control life and death. They're also involved in helping to recycle components, temperature control, redox. There's probably many others that we haven't actually sort of mentioned here. And one of the central components, obviously, is the Krebs cycle. And we won't go too much into that today because this gets really detailed biochemistry. But the point about this is it's like a roundabout which can go in either direction. And one direction it can help with energy, other directions it can help produce metabolites, and it crosses over with inflammation. And as it turns out, a lot of disease processes, including COVID and certainly cancer, seem to manipulate this, resulting in metabolic reprogramming, and which can be detrimental. And of course, the crossover here is we now know that certainly when astronauts go into space, they start to display metabolic disturbances, and potentially, as Ashvin will talk about, the mitochondria are probably pretty, they're implemented in this. And of course, there's also quite an interesting crossover here with the aging process, which we'll talk about briefly. So what are the key things to think about regarding possible life in space? One of the concepts that's been emerging over the discussions we've had over the last year with various people, and more recently with folk talking about the origins of life and certainly space, is that life obviously evolved on this planet, and it can be described as a self-organizing, far from equilibrium, dissipating negentropic structure, which is what Schrödinger called it. And key in understanding of thermodynamic principles, including entropy and the sort of urge of the universe to equilibrate everything. And certainly there may well be a role for quantum mechanics, which is what we're very interested in. And so did life emerge because of a particular set of circumstances on Earth? And does it still rely on those circumstances or signature of the Earth? For instance, we certainly know that water is important. Chemistry is obviously important. There's geochemical chemistry, which was around well before life came along. Energy gradients. And today, I'm assuming one of the strongest theories, which is related to the alkaline thermal vent, which gave rise to a proton gradient and eventually chemiosmotic energy capture. There are other theories available. I will point this out. There's a solar potential theory, there's the RNA theory, there's the fatty world theory, there's all sorts, but we kind of quite like this idea of the thermal vent. And key in this would have been the right kind of stress to stimulate it. Without stress, nothing would have ever evolved. This is a fundamental principle of evolution. But what about other things like electromagnetism, gravity and gravity wobble, as Steve Thorne talks about? And so how do we go from here over to here? And what happens when we do? So what have we already learned? Well, we know, and this comes from our background, we certainly know that a poor lifestyle accelerates the aging process. If we don't move much and eat too much, we get all sorts of metabolic problems, and it seems to lead to an acceleration or development at a younger age of a great deal of the many more unpleasant diseases ranging from cancer to heart disease, diabetes, you name it. And we also know that lack of gravity seems to cause many of us similar problems, not entirely the same, but similar, and could, for instance, lack of gravity accelerate the aging process, just as a thought. And we also know from early research that the lack of the Earth's magnetic field can result in embryo changes. So things which are growing do seem to require some kind of magnetic field around them. We also know from quite a few studies now over the years that you can manipulate metabolism using electromagnetism, and certainly to transcranial stimulation, but certainly and also in wound healing and things. There's a lot of research going, so we can manipulate biology using electric fields. And we also know that health is very strongly tied to circadian rhythms. And when we start looking at the biochemistry behind circadian rhythms, it's very strongly linked to aging and indeed gravity as well, and inflammation. So there's clearly something very important in there. And we know that astronauts, even if they're very fit when they start, do develop some metabolic problems quite quickly. Now, sorry for this complicated diagram, but this is basically a point we're trying to make, that if we think about the origins of life, what was essential or what was needed at the beginnings of life? And this is an idea we've published. It may or may not be true. But the point we make is if you think about the thermal vent, in the thermal vent, there was a massive flow of ions, and ions obviously are moving charge, which create fields. Were these fields important in shaping life? And of course, this leads on to all the way down the road to Mike Levine's work in morphogenetics. And as Mike has given some brilliant talks on showing that, the field around life could be quite important in dictating its shape. So what's going on here? Could this be an imprint of a signature that it needs? And this is one of the slides from Steve's presentations. But clearly he makes the point and links it to this idea that actually it may not just be, say, the movement of the circadian rhythm related to light, but it could even be a link to the wobbles in gravity caused by the movement of various bodies around each, like the lunar or the solar cycle. And one of the interesting things that come out of this, of course, is this idea that we start to think about circadian synchronicity, and is there something else going on other than just light? And one of the topics we had quite recently was talking about chirality. Was the rotation of the Earth and vorticity and the Coriolis effect important in, say, for instance, which way an ATP turns, or which way molecules tend to develop, being left or right-handed? These are big questions. We don't know, but they could well be quite important. Sorry for this other diagram. This is from an early paper we did, but it basically explains this idea of canalization. And this is the concept that during evolution, life will have evolved to live at its optimal state in a particular set of conditions. And in this point, we make the point that mankind over the last 100 years has moved from living quite often in a state of either feast or famine to a constant state of feast. And this is an idea by somebody called Stoger, which is we get canalized at a certain point. This ball represents our sort of like our metabolic health, as it were. And the width of the bowl represents our metabolic flexibility. Now, we know that if you famine a bit or exercise, you get better metabolic flexibility. This bowl gets slightly bigger, so you're better adapted. But of course, through the generations and through epigenetics, it seems also, and familial social things, we're pushing ourselves further and further outside this normal, like what is effectively like a flight envelope. And if you're healthy and strong and do the right things, you do better than if you don't. And so, of course, what does this mean to take-home messages? Well, we come from and I work with Jimmy Bell at Westminster on this idea of optimal health, and defining what optimal health actually is quite difficult. We talk about normal, but actually what is optimal health? And somebody quite recently has published a paper looking at what is healthy in the American population. And when they actually look at various metrics, they actually found that only about 16% of Americans actually fit the bill of being metabolically quite healthy. The rest are not, which is quite telling. And especially if this goes through generations. Basically, what it seems to be is that, in terms of pinch a term from the aviation world, it seems that if we evolved inside our flight envelope and it was generated by our environment, but if we start to go outside of it and we can't adapt, we seem to age faster. And if conditions are too extreme, of course, we don't do very well. We don't survive very well without oxygen, for instance. So does space push humans outside of this envelope? And clearly, potentially starting metabolic flexibility, which is quite strongly linked into the mitochondrial function, it suggests that we may well need to take our own signature, Earth signature, with us. And this is just a little ditty showing that if you're quite fit, you have, most astronauts are very, very fit when they start, but they start to develop problems, particularly with their mitochondria. And we know that over intermediate time, there are problems. What happens in the very, very long term? And the interesting point falls out of this. What if you put somebody who's not very fit into space? How long is it before they get very unhealthy? And so the idea being here, we need to apply whatever it is that Earth is providing us, we're coupled to help us survive in space. And so in the final slide here, before I introduce Afshin, in space, no one can hear you age, but your metabolism doesn't lie. So something's going on and we need to investigate it a bit further and understand it. So with that, I'll hand over to Afshin.
Thanks for the introduction and thanks for having me here to give a talk. I'm excited to be here. Just a disclaimer. Well, one disclaimer is I'm actually a physicist by training, but 20 years ago I switched over to biology. So my quantum physics has been it's pretty rusty. So other than that, in my talk I'm not going to be really talking about the quantum aspect, but I figure that's what we're going to discuss after the talk. But basically looking at the mitochondrial dysfunction being like a universal driver, with space flight, and how gravity and the space radiation primarily are going to be the two main factors that are going to be driving the space related issues. So this is a background. I'm sure some of you might be familiar with space flight experiments and resources available, but I thought a background overall would be nice for everyone here, just to know what's this involved with space flight experiments. So why care about space biology research? I think Alistair did a nice job to bring up to why we should care. But one thing is that Alistair pointed out also is that space is an accelerated model for aging and mitochondrial disease, and you'll see why when I start presenting the talk and the biology of it. So that's one of the main fundamental things where, when you do space research, it's not just for the 8 astronauts or the few astronauts, coming up in the future to be in the past, go into space and figure out safe for a small population of humans, but the research that comes out of it, because space has the platform for accelerated aging research or accelerated mitochondrial disease, is actually you could get a lot of data that might help clinically, because within a month span you might see the damage done to the mitochondrial, which might take a year or a decade even on Earth. So in a sense, I say if you could cure it in space, you could most likely have the cure for the clinic here probably pretty easily. So future explorations are, as you saw today, Artemis went up. This is from an older article, but Artemis went up today and this morning. Sorry, let me go back. And so that's the next. We're going to the moon, and then eventually, like in a few more years, I think they're going to start building a moon base and have Gateway where it's be floating around the moon, a station, and then also people will be establishing bases on the moon, subdue research and figure out ways to make it safe for humans to travel, and then eventually go to Mars. That's the whole goal, where they want to have humans go to Mars and establish a colony there, and then obviously long in the future is to future explorations to see things go further out in the galaxy or the universe, like Europa. So kind of like Star Trek in a sense, right? Explore the stars. So as I hinted at earlier, the two main space, there's a lot of hazards in space. There's one review we put on Cell a couple of years ago, discusses some of them. There's, for example, distance from Earth, the small confinement you get, the hostile closed environments, which can produce things like antimicrobial resistant bacteria, which I won't talk about. But the main two components that I'm talking about impacting mitochondrial is gravity, or the lack of gravity, and then also radiation. Space radiation specifically, which I'll go into in a second. But just to give you an idea, this is in this plot here in the lower left, let's say the annual is going in the middle bar. So the abdominal CT scans you might get on Earth is about here for the amount of radiation you get. But if you, let's say, go to ISS, which is in low Earth orbit, so this is one thing to keep in mind, that the ISS is in the low Earth orbit, which you guys are probably familiar with, and it's in the magnetosphere, so it's protected by the Van Allen belt. So that would, as you guys would know, reduce the amount of radiation that might astronauts and other experiments we do in the ISS they would receive. Once you go to the deep space, now you're getting the full-blown deep space radiation, which I'll go into in a second. So this is kind of equivalent here. This one here is like in a 6-month mission to Mars. And the equivalent to CT scan is a lot more damaging. And the other part that you have to keep in mind, the type of radiation is a lot more damaging, which I'm going to get to in a second. So I just briefly already mentioned that. The moon, the Mars, you know, is the next 2 missions. And of course, the lack of gravity and the space radiation I'm going to get into. So there's 2 types of space radiation, as I didn't mention, but you saw the movie. One is the solar energy, solar particle events, it's called. So basically a high dose of protons at different energies the astronauts can be exposed to. Those are more predictable because you can see a solar flare happening, and then astronauts could try to reduce somehow the mitigate the damage that might be caused by it. But of course, there's no shielding that would protect you against it. But they could take some precautions. The other part, which is constant, is the deep galactic cosmic rays. So galactic cosmic rays, which is the deep space radiation, which is basically radiation that's evolved, is traveling through the space that comes from black holes or supernovas and so on and so on. So the majority of space has protons and helium, small ions, and then it goes up to higher ions. So back in the 50s, they even knew that space radiation can be damaging, and then of course they can result to maybe potentially causing some damage to people, maybe turning into a superhero. But more realistically, I think Marvel got this wrong. I'm going to show you that these people probably would have been in hospital instead of becoming superheroes. But just to give you a little brief background of the different types of radiation, on Earth, we're exposed to low LET radiation, which is basically gamma radiation. So these are basically high moving charged particles that go through your cells and scatter, once they go through your cells and travel through your cells. So in a sense, it's like sand particles that's traveling in your cell. High LET is the basic type of radiation that's in space. And this is particles, ions ranging from, this chart shows from like iron particles to small things like helium and protons. And high LET is, they're slower moving than the low LET radiation, but they deposit all their energy in these cells. So LET, I forgot to mention, is called linear energy transfer. So it's the amount of energy the ions or the radiation will transfer to the cell. So high LET, in a sense, is very damaging because you can think of a bowling ball, like an iron particle, is a bowling ball that goes through the cell and also deposits all the damage right in the middle of the cell and causes all that damage. So an equivalent dose, let's say of 2 gray of gamma, is not equivalent to the same as high LET. So, as I said, when you go to Mars and come back, it's estimated you might get 0.5 grays of galactic cosmic rays, so a combination of all these ions. So this is estimated to be equivalent to something like 10 to 15 grays of gamma you might get on Earth. So as you can imagine, it's a lot more damaging. Although going to Mars and back is a slow dose rate, so it's a slow accumulated dose that you would get eventually 0.5 gray. So what would this cause to health risks? So some of the health risks, we again summarize in this review article, are some of these health risks are dependent on sex, as we highlight here, but a lot of them are, independent of sex. So things like renal issues, concern, kidney and liver issues, and CNS, circadian rhythm issues, as Alistair pointed out, can be an issue. And everything, you could think of, like muscle degeneration, bone loss, cancer risks is even a long-term issue. And one thing also, there's an ocular issue called SANS, space-associated neuroocular syndrome, which astronauts tend to start declining in vision, depending on, not every astronaut, but depending on how long they're in space. And this chart here shows, like, in deep space, what we categorize could have potentially more risk than, lower Earth orbit missions or compared to other kind of risks. So, going to space, our job now, and all this research we're doing, is to figure out countermeasures to help make it safe, so that, let's say, if the astronauts go to Mars, they actually can function and be healthy, as opposed to by the time they get there, within a few hundred days, 300-400 days, that they're not going to be actually sick with many things and they can't even function by the time they get there. So the type of experiments now we can do to actually come up with countermeasures and determine the biology is, one, send samples to space. So we got our mousternauts here. There's actually videos of mice that were in space. They seem to adapt fairly well. They float around, and some of them just whiz around. They don't need a little mouse wheel. They could actually do that in in gravity fairly well by themselves to exercise. And I'll show you in a second that, you know, people just don't set up mice, but other organisms too. So now let's say if you don't have access to payloads to go to the space and grants to send experiments, you could actually do experiments on Earth. So one way to do this is do a simulated space radiation on Earth, which I've done, and I'll show you some of the data on that, is going to Brookhaven National Lab, which is a high energy physics collider at Long Island, New York. And NASA in the early 2000s had set up a NASA Space Radiation Laboratory where they took part of this beam. Now, here you can see the beam when you can put samples in front of this. So now you could actually irradiate your samples with the heavy ions that you see in space, like iron particles, helium. And now they have this galactic cosmic ray mixed beam that simulates what, you know, an astronaut might get when they're in deep space outside of the low Earth orbit. So that's one way to do it. It's the only place in the world that really has a facility that you could actually do experiments, mice, cells, whatever you want to put in front of the beam. And one way to simulate microgravity is these hind limb unloading, it's called, setup where you hang. It's kind of silly, but you kind of hang the mice by the tails, but it's the best thing we have. And this simulates after a couple of weeks the unloading of the weight from the back leg. So now, you know, you get muscle degeneration and bone loss and other factors that mice or humans and other organism might experience when there's a, you know, microgravity setting or lack of gravity. Again, this is not, both these simulated ones will never be truly capturing spaceflight experience, but it's the closest we're getting, and it's the best we have. And you get some interesting biology that correlates very well to what's happening in space. And then there's also other newer models by Stuart Rudcove and Mary Matrua. That's like a partial weight-bearing rat model. It's kind of a picture of how they do it, and where they have a harness and they can, you know, lift the rat in different capacities to simulate different types of gravity. So like the gravity on the moon, of course, is different from when you're in deep space, and the gravity on Mars is going to be different from the moon and other factors. So they have a way to simulate maybe the percentage of gravity there. Again, it's called the partial weight bearing rat model, and you would simulate the unloading of weight within the muscles. And then, you know, I'm talking about rodents, but there's other types of experiments which I don't do, but we have collaborators that do nice experiments. And of course, every one of these models, you could represent or could be comparable to a different human model, which you guys are all familiar with. So this Drosophilas, they use fruit flies and they send them to space. These type of experiments you can do. There's also C. elegans they could send, which is good models they could have some comparable CNS and muscle kind of comparisons you could look at. Even zebrafish they've even sent squid. And of course they do plant research, which I'm gonna briefly mention in one slide, but and show you mitochondrial function even in plants. But one thing is, for plants, that, you know, the research they do is they want sustainable food and sustainable growth of agriculture. So astronauts, when they travel long distances, they could actually grow their own food. So that's why they do a lot of plant research in space. And there's a whole group of people working on astrobotanists, as we call them, they're working on. And of course, there's microbial research and then in vitro cell culture. So one maybe aspect, if you want to get yourself familiar with what's happening up recently in space biology. A couple of years ago, Chris Mason and Susan Bailey, we put a large package of papers out in collaboration with Cell Press. And this had about 39 papers in there from various aspects of space biology. So this might give you a more up-to-date version of what's happening in space research if you want to check that out. Stay tuned. In March, we're going to have another package coming out with Nature Press family, where it's going to be focusing on more human aspects, because it's going to have Inspiration 4, which was a privately commercial run mission last year, and also JAXA, Japanese Space Agency, data focused package. And there's going to be a bunch of other companion papers associated with that. So stay tuned in March for a release of that for the more up to date, 2.5 years later, papers coming out. So one of the papers that came out of this was this mitochondrial paper, which we're going to talk about. Doug Wallace was also an author on this paper. These covers were actually from Scott Kelly, one of the twins from the twin study data. He took these pictures and the cover. But so I'll be discussing this briefly, because you could probably read the paper for more, but then I'm going to go into more of the newer data and the newer research I've been doing. And then there's another aspect I'm going to discuss is microRNAs, as Alistair pointed out. I work on microRNAs, but how microRNAs are related to mitochondria influence them in space. And these papers we put out in the package, but from the basis of this, I'm going to show you newer research on that. So one resource that, let's say you say I don't have any money to run any of these experiments, can I analyze data? You can actually go to GeneLab, which is a NASA resource that has been developed, so all the omics related to space biology research ends up here. So it's free. Anyone can go here, look at the data repository. They even have a portal where they have visualization and ways you can analyze data on there. If people weren't bioinformatically inclined, they could actually just look up their favorite genes and say, does this actually change with some data sets and tissues, for example, they're looking at. And one way people can get involved now is these analysis working groups, which we have for our Gene Lab, and I lead the multi-omics systems biology, which has about 250 plus people now in there. But there's neat little projects that scientists from around the world work with each other. This cell paper here actually came out of that group, and it's a large collaborative group, and that was one of the nice papers and impactful papers that this group works on. But there's now a bunch of different projects, and one project I'm talking about is now taking that paper into evolving it into more in-depth mitochondrial research and how space is related to that. So as I said, we're going to talk about this impact in mitochondrial space first, and then switched over to microRNAs, how they impact the mitochondrial space. And this paper here was, of course, not just me, but as I said, it's an analysis working group. There's some people outside of analysis working group, some people you might recognize here. So there's a lot of people involved with this paper, and it evolved to be a very nice paper, and it's a large collaborative work. So the way I approach work is the more the merrier. So the more people we have involved, the better it is to get some really nice research out in a quick and concrete way. So the overview of this paper basically was, we all came in the group and said, my idea was, is there some sort of master switch that turns on or off when you're in space biologically, whether it's due to, the magnetic field or the space radiation or the gravity, it's all synergistic in this case when the astronaut's in space. So what happened is that then we go into the Gene Lab, mine the data from mice that were flown to space from 2 different strains, and they looked at all these different organs. There's also in vitro data, cells sent to space, and even some hair follicle data from this JAXA study is available in Gene Lab. And then people, depending on their expertise, went to look at the transcriptomic data, or methylation data or proteomic data, and even there's some metabolomic data to look at the metabolites. So then when everyone came back independently, so it's completely unbiased, and they said, okay, let's see if they might find any interesting top hits or top factors that are popping up in their analysis. Everyone came back after a while and said, independent of the organ or the cell, mitochondrial was always a top hit. And in the past, people would have always ignored that. I'm sure mitochondrial researchers have dealt with that for the past few decades, have agreed that a lot of people might ignore mitochondrial functions. And that's why previously there's been some inklings of papers coming out pointing to mitochondrial dysfunction in space, but never before this had anyone really highlighted the fact that people should really pay attention to mitochondrial function in space, because it's a universal dysregulator in basically all your organs and cells, whether it's in vitro studies or that. And then eventually what we did was, with collaborators at Johnson Space Center, we got astronaut physiological data from about 60 astronauts from the past few decades, related with mitochondrial markers. And then also from the NASA twin study in collaboration with Chris Mason, from Scott Kelly and Mark Kelly, then we got human data there. It's an N of 1, so you take it with a grain of salt, but at the same time, it's kind of a proof of principle. You can see that indeed the mitochondrial dysregulation is translatable to humans. It's not just a mouse phenomenon. So there's going to be, I'm going to quickly go through this paper to get to the newer data, but don't really get bogged down with these hairballs and the heat mass, but I'm going to tell you the overall theme. So one thing is here in panel A, in this paper, people went in to look at in vitro data that's there. So these cells that were sent to space. And overall, the unifying theme, the one that pathway, this is from pathway analysis, or the genes that were universally dysregulated, no matter what cell type you looked at, was all related to mitochondrial dysfunction. So which is interesting there. Then when looking at from the mice data, these are from 2 different strains of mice, and this comes into play a little bit later, but there's C57 mice that were sent to space and BALB-C. BALB-C mice tend to be more radiosensitive, and the C57 are more radio resistant. So this comes into play a little bit later to see the interplay also from mitochondrial damage that might occur in the 2 different mice in space. From the newer research, we didn't discuss this as much here because we're looking at independent changes regardless of strain or sex. And indeed here was mitochondrial dysfunction. And these hairballs here and nose are basically looking at both the proteomic data, which is the inner circle, and the outer circle is the transcriptomic data, and the wedges refer to like the different tissues and the strains. And then here's related to just the main organs, the liver, kidney, eye, adrenal gland. And here's the muscle related factor, which I'm going to show you the differences between the muscle and the organs in a second. And this methylation data related to that. What's interesting now, mapping all the mitochondrial genes from mitochondria onto these different tissues, we see that liver indeed has the highest mitochondrial gene dysregulation here. And then for the muscle, the soleus muscle, this is what this orange color bar is, has the highest dysregulation mitochondrial gene, significantly regulated. Interestingly enough, soleus muscle is one of the first muscles to get impacted with muscle generation in space, that part of the muscle. And we know from, I'm sure everyone's familiar, how the liver and mitochondrial pathogenesis occurs with liver type diseases too. So let's go on to the next slide. So then to further look at, how data is, again, looking at mouse data, we see that this is from metabolomics done on two different muscle types, the gastrocnemius and the quadriceps from the mice that were sent to space. And one of the people in the group did metabolomics on this, and when they came back, they saw that there's indeed a lot of top hits are related to mitochondrial structures of the blue font, but the ones that are not in the blue font are also all indirectly related to mitochondrial function. I'm sure everyone looking at this will know these pathways like glycolysis, lactose secretion, all these metabolic pathways that are indeed also related to mitochondrial dysfunction. So everything that popped up here was pretty interesting to see. We looked at different things related to OXPHOS and integrated stress response, which we'll go into more detail now for the newer projects. When you look at the twin study data, Scott Kelly and Mark Kelly, it's interesting, this is PCR down on the mitochondrial DNA, some factors there. And you can see here's the ground, the HR means the ground, and the twins, the twin on the ISS. So Mark Kelly, for example, did not really change much with their mitochondrial DNA here. But when you look at Scott Kelly over time, I think this time point is about, if I remember correctly, it's about close to 150 to 200 days in space. You see a big spike happening in the mitochondrial DNA. And then the orange section here is when they come back post-flight. Luckily, some of this stuff comes back down to normal, but you can even see here, the last time point, I think, is 100 days or approximately after flight. You can still see some of these things are very high up. Now, when looking at physiological data from astronauts, from the blood and urine factors that they have measured in the past that are related with mitochondrial, so we got antioxidant capacity, and then you got these two factors in urine, prostaglandin and 8-OGDH, that you see that in fact these markers are really cellular oxidative stress. You see that indeed, in the urine, these are. Let me back up there for a second to explain the x-axis for you. So the L is launch, 45 days before launch. So this is astronauts before they flew. FD is flight during flight, and these are the time points during flight, so 15 days, so 100 days, and R is return. So 0 means the day they came back, they got the blood, 30 days after flight. So then you can see indeed, like for example, things with antioxidant capacity from the blood, there's a significant reduction, but luckily after 30 days, it comes back to normal. These other factors related to oxidative stress in the urine, some of these things, again, they get highly upregulated when they're in space from the urine, and then when they return, they start coming back down to the normal levels, which is good to see. But indeed, constant exposure of it in space, you're going to get this elevated, accelerated, as I call it, model for a mitochondrial type diseases occurring constantly. Luckily, it seems like some of this stuff returns back to normal when they come back to Earth. What's interesting here, if you look at this right plot here, is from the NASA twin study data, is the mitochondria genes again, all the 1137 genes mapped onto what happens to Scott and Mark Kelly. And I know there's a lot here, but the overall theme is this is comparing these top bars, comparing like pre-flight versus ground, in-flight versus ground, post-flight, and so on. And then you got in-flight versus pre-flight, comparing like the black comparing to Scott Kelly himself over time. What I want to point out is these are the different cell types from the blood they looked at. And what I want to point out is that indeed over time in space, where this comparing in-flight Scott Kelly to himself, the mitochondrial distribution, which just shows, changes, the profile changes. So that's the whole kind of theme I wanted to show here. And indeed it does. You see that over time, depending on the cell type, some might have more profound effect, as you see here. But if you compare over here, the whole distribution changes for their mitochondria. Scott Kelly does compared to the ground. And this is some nice analysis done by Deanne Taylor and Jimmy Mann Kim from CHOP, and they did, this is called metabolic flux analysis. And this is taking the RNA-seq data and use some modeling technique, which I won't get into, you can read in the paper, but you can actually now predict metabolic fluxes based on transcriptomic data, RNA-seq data, which is pretty interesting. So this is now looking at the liver and muscle interplay that I briefly hinted at in the previous slide. And what's interesting is that, you know, this is the liver data related to mitochondrial metabolic flux activity, and this is now muscle data here, as you see here, the color scheme. So you can see in the liver, definitely, this is the ground control, and this is the space flight. So in the ground control, oh, sorry, reversed, space flight and ground control. So the blue is the ground, this is space flight. And so what you can see here is that indeed the liver, the mitochondrial or metabolic flux activity related to mitochondria in space, they're heavily upregulated in mitochondrial activity. This is a common theme we see over and over again. But in the muscle, due to the the muscle degeneration that's happening, the mitochondrial, this is space, this is ground, the mitochondrial activity, the fluxes are suppressed. So that I know in the literature, people, I think, so in the literature, what people have seen also related to muscle degeneration is in sarcopenia, that this common thing happens where the liver is trying to overcompensate, or the hypothesis would be here, the liver is trying to overcompensate the mitochondrial damage caused in the muscle, and this interplay happens. And the downstream factors are now, you know, for the mitochondria, it shows like lipid and fatty acid dysregulation. We know how, I'm sure everyone's familiar with mitochondrial dysfunction and lipid homeostasis. So I won't get into that here because you guys are all mitochondrial experts. But so there's a, you know, interplay there. This is again the transcriptomic data. What's interesting with the, again, the astronaut physiological data, you can look at the cholesterol levels and LDL and HDL, again, before flight, during flight, return. You can see elevated significantly amounts of things related to cholesterol, LDL, HDL. Again, as Alistair pointed out, these astronauts are the most top 0.001% fit of the population, and they get increase in cholesterol, HDL, even though they're very active on they get a 2-hour regimen of exercise on the ISS, and they're very active all the time. Their diet is well regulated. And these factors not necessarily come back down either. Some of them stay up for cholesterol even 30 days after space. And again, you see that interplay here where this is the liver, this is the muscle data. The muscle shows heavy suppression of these activity for the metabolic activity, flux activity, while the liver is upregulated. So this is another paper related with the skeletal issues. Again, and then, you know, things related to circadian rhythm is downstream of this, which Alistair again pointed out in the introduction. We see activity happening there, cell cycle control being dysregulated again, and then also things related to renin, which is you know, related to mitochondrial. So then briefly, this is what we discovered in the paper, being a universal regulator. And now what's evolving within the group is now looking at future mitochondrial changes that might occur, and more specific analysis, and how this might be related to all species. So this is now a project, as I mentioned, the analysis working group. These are some of the people who are leading it. You might recognize some of them. And what's interesting now, how this evolved is saying, okay, we came up with this rodents and humans. Can this really, is this mitochondrial dysregulation universal? And in collaboration with Richard Barker, who's at the University of Wisconsin, he's a plant astrobotanist, as we call him. And after he saw what we did, he went and looked at the plants. And I guess in plant biology, mitochondrial is even ignored more usually. But when he did some nice adjustments in his analysis, he actually saw mitochondria as a top function and top dysfunction, I should say, being dysregulated in a lot of the plants that were sent to space. And this is some of the initial analysis, but I'm going to go to more in-depth analysis of the mitochondrial pathways. This is in collaboration again with Tin's lab and their graduate students, Negar and Fee. And again, now here we're looking at they're using a tool he calls CPA, which is similar to, if you're familiar with gene set enrichment analysis, so kind of pathway analysis, but it's a more evolved tool that they've created, which is remove some of the bias you see in regular pathway analysis tools. So what we did was take the mito pathways, which are mitochondria defined, and now they're mapping it to different factors. Now this is some mouse data looking at, it's hard to read here, but I'm going to go to the next plot to show you some separation of this, but looking at different tissues again, and there's more recent data from Gene Lab now on there, which wasn't in the original paper, like thymus and other factors. So let me go to the oxphos-related pathways. So again, so this is Rich plots looking at this, and just to give you orientation, these little red bars here and the blue bars associated with the data set, and the Rich plots, if you're not familiar, anything to the left, let's say if this is positive here, see if you look at the enrichment scores, negative means this is downregulated, or positive means upregulated. So this little red bar here, anything to the right of that is significant. Anything to the, to the left of that is not as significant. So it shows you how significant maybe potentially the genes associated with these pathways are with each of these. And what's interesting now, the color scheme above, these are now where the BALB/C and C57 mice come into play. So depending on the data set, one's a C57 mouse, one's a BALB/C. Bone marrow, you just have one type of mice there, and that's C57 mice. But one striking thing, for example, the eye. You can see the BALB/C mice are here in the blue and C57 are in the red. And they clearly have a distinct different OX-phos pattern in the eye, as opposed to, and remember, BALB/C are more radiosensitive, and C57 are more radioresistant. So this difference in mitochondrial changes, I think, should be attributed to space radiation more than microgravity, based on that. But I'm sure there's microgravity component bringing up, the differences due to their genetics here too. And something that's interesting, for example, in liver, as you see here, this is from 4 different missions, and they're combining C57 and BALB/C, and also earlier shuttle missions, so the duration's shorter, in space in the ISS. Typically, these mice are there for 30 days in space. Some of them are a little longer, and some of them are a little shorter, but plus or minus like 10 days, about 30 days. But you can see the liver, as we showed in the previous paper, indeed, it's no matter the species and the mission, it's always mitochondrial OX-phos activity is increased. As opposed to some of these other ones, you can see like the kidney, again, you see that dynamic where BALB/C is the blue one, C57 is the red one here. That reverse dynamic. Thymus is only one type of mouse. C57 is heavily suppressed for the OX-phos activities, and skin. Again, these are the 2 different strains of mice. But that's interesting to see here interplay. And again, for this work that we're doing is ongoing work, and the more collaborators, the merrier for this. So people want to join this project, it'd be interesting to have your input. These are some Sankey plots, again, now looking at some of the data. This is just a brief snippet of all the data we're looking at. With these mitochondrial functions, we've actually done it for all the metabolic pathways. We're working on the mitochondrial central dogma ones, and so on and so on. So we have more data than we could do with, and we're going to be regulating, going over the data and putting processing for the paper. But this is just an example from the OX-phos. It was easier to show here, the preview of what's to come in the paper we're preparing. But these Sankey plots now show sort of the distribution of, this is from the mice, for example. You look at all the different tissues, the strains of mice, and then the thickness of the line is how many genes are associated with, each of these connections you make. So you can look at, for example, from the mitochondrial central dogma associated pathways according to mito pathway, what might have more influence over others, depending on the data set you're looking at and the muscles. What's interesting about plants now, as I mentioned, we have plots like this related to plants that are showing a similar type of behavior of mitochondrial pathways. And this is just a Sankey plot looking at plants, looking at different types, areas from the plant, like the shoots, the roots, and then how they're related to mitochondrial dysfunction. And overall, the theme of it, what we see is that indeed the plants have similar behavior to humans and mice, and some of the other data we're looking at with mitochondrial dysfunction that happens, like the OX-phos activity is similarly behaving than mice and humans. So, and other factors of metabolism. So the plants might not be so far behind with mitochondrial dysregulation as to mice and humans. So this was interesting to see. And then I want to show in the last, I guess what we have, last 15 minutes or so, how microRNAs are also a key aspect of impact in space and how it's related to mitochondrial damage. So that, starting from this paper, where we developed, I'll show you briefly. I'm not going to go over this paper in detail, but I'm going to show you the work that's ongoing and the papers we're preparing now to show. But in a nutshell, what we identified a group of microRNAs associated with spaceflight signature, and then we figured out it could be a good biomarker in a way. If you inhibit them, might be a way to as a potential future countermeasure. So what are microRNAs? Why study them? Why are they important? I'm sure most of you are familiar with them, but classically in biology, this is what we knew what was going to occur. But 1993 was the first time someone discovered a microRNA, but early 2000s is when it started taking off. And what they discovered is that indeed these microRNAs that are 22 nucleotides inside can regulate hundreds and hundreds of genes, not just 500, but I should update this. Due to their size and stability, they can be found floating freely, or freely in quotes, in every fluid you see. So there's a few different ways they can float in your body, where there's floating with a protein attached, an agroprotein attached to them to make them get in and out of cells, or they could be packed in exosomes or lipids. For me, distinguish between them, between the different ways they could be delivered, because I'll just look at how they might be, regardless of what they're packaged in, they're still in the blood and can be used as a biomarker and inhibit the same way. They're involved with every different aspect of biology, from transcription, translation to even DNA interaction. They're known to be involved with all aspects of disease and even healthy. Of course, the healthy microRNAs associated with healthy functions, and also microRNAs with diseases. And they're not only found in mammals, but every living organism, highly conserved over species. So it makes it a good model. So how could microRNAs be related to mitochondria? There's been literature in the past decade or so that shows that mitochondria has heavy influence, and some microRNAs can influence mitochondria in a good way and bad way. And these are some papers associated with that. So a lot of times in disease like cancer and stuff, it's evolved where it show, and heart disease, where some microRNAs, when there's impact or that disease progressions, could cause damage to mitochondria. There's even been microRNAs that show that come from mitochondria called mitomeres. And basically it shows that these mitomeres are, mitochondria can use these mitomeres as a way for it to function right and behave, the mitochondrial functions you want them to behave optimally. But, if there's damage done, mitochondria releases some of these microRNAs that might further, enhance mitochondrial dysfunction. So this is, again, some other paper and some other literature looking at different microRNAs associated with that and how it might impact different factors of mitochondrial coming from the mitochondria. So in these, again, these are the papers that originally came out looking at the microRNAs I characterized. This is a very complex summary figure from this mitochondrial or microRNA paper. In these papers, I didn't talk about the mitochondria, but this is the evolution now for what we're working on now. But overall, what we show, as I said, in this paper, we show that the microRNAs we identified was indeed circulating in the blood from mice that were flown to space, simulated experiments we did on Earth, and even from the NASA twin study data. Now, what's more exciting part of it is that can you use these microRNAs as a therapeutic or as a countermeasure to mitigate the damage caused in space? So we started with using a 3D microvasculature model, which was in these papers that we put out a couple of years ago. This 3D microvasculature model was created by Pete Grabham at Columbia University, and it's basically from HUVIC cells derived, so a human umbilical vascular endothelial cells. And there's no blood flowing in there. It's just a simple, microvascular endothelial type 3D human tissue model. And what happens is we said, okay, and in space, when you have these 3D tissue cells, when you radiate the space radiation, they collapse. So we said, okay, can we find a component of these microRNAs which identified these 3 microRNAs to be associated with cardiovascular risk associated with space flight, and then see what happens if we actually inhibit them? Can we rescue the damage done? So we went in Brookhaven, and we radiated these with these microvascular 3D microvascular tissues with the galactic cosmic simulated beam. So the Brookhaven has a simulated beam where it's protons at different energies, as you see here on the top and bottom, with heavy ions that come in the middle of that. So it kind of simulates what happens in space, because the idea is you will get a black background of these light ions, and then eventually you're going to get once in a while a heavy ion going through the astronaut. That's what you would see. So, 0 gray, you see this is these microvasculature nicely organized, but then at half a gray they start collapsing, and then this is a quantification of that. And then this is also the microRNAs that are being overexpressed, and these are the other microRNAs that have, part of my signature also overexpressed when you quantify that. But these are the ones specifically I said would be really the microvasculature effect. So then if we inhibit these, can we mitigate this damage? So now we see what we did was take these integomeres, which is like an antisense or the antagonist sequence to the microRNAs, and with Vinu at Life Tech, he's developed these self-delivery type integomeres it's called FANA technologies, where it's kind of a self-delivery lipid delivered system, which I won't get into, but you can basically then sprinkle in your cell culture, get in and out of cells, find the right target, or you could inject the mice show. So we injected that we put that in the cell culture 24 hours before radiation, waited 6 days, and lo and behold, to our nice surprise, we completely mitigated the damage, as you see here, caused by the space radiation. This is a scrambled version of the vehicle. It's a scrambled sequences to make sure the vehicle doesn't contribute to this in any way, and you can see that it doesn't. So that was really exciting to see. We did this. There's 2 different models he has. He has one as a mature version, which is here, and then he has one we call developmental angiogenic model, where starting to develop the vasculature. So there's buds that form and then spread out. And the same thing happens here. When you radiate them with the galactic cosmic rays, the buds actually get inhibited from forming, and they kind of form a collagen layer around them. so then it doesn't form the buds eventually in the vasculature, doesn't become, or the tissue model doesn't become mature anymore. But again, when we applied the integumentary, indeed it rescued it very nicely. So how are these microRNAs related with mitochondrial? And there's been literature that's shown that these might be potentially mitomeres. So there's MIR125B, there's papers that show the interplay between mitochondrial metabolism. There's MIR16, again, shows this dependence on mitochondrial apoptosis if it's overexpressed, and LET7A is another one that shouldn't be overexpressed, it's bad for mitochondria if it's overexpressed. So you can see the damage caused and causing mitochondrial, basically apoptosis or other dysfunction related to that. So then now we've evolved to say, what's the biology behind this? So we've taken this further now in the papers we're working on to submit at the end of this month. And this is, again, the same type of experiments repeated many times. So this is very highly reproducible. One thing we did is, if you don't inhibit all three microRNAs and once doing it by themselves, what effects might produce inhibitory effects? And surprisingly, we see that even by themselves, it's just here, this is your sham, no treatment. This is your radiation here without any treatment. And this is radiation with either 3 integumentary given at once, which we originally did, or by themselves. This is your mature and angiogenic model. And you see that by themselves, they actually seem to have some rescuing properties. So we have to further look into, maybe you just need 1 integumentary instead of all 3, or maybe all 3 is still, we need that. So now we like looking at the biology. So one thing we did was, with radiation biology, if you have radiation damage, you cause DNA double strand breaks. So how does this integumentary treatment might impact DNA double strand breaks? So first of all, do microRNAs regulate DNA double strand breaks or are involved with that? From some mouse experiments we did radiated with galactic cosmic rays. 24 hours later, we had sacrificed some mice, related to going to Brookhaven and radiating the 0.5 grade GCR, and then looked at different tissue from liver, soleus muscle, blood, and heart. And then we basically did some microRNA sequencing on them. And indeed, we see that globally, there's a lot of this. These are all DNA double strand break repair pathways, specifically we looked at for different pathway analysis. And indeed, we see a lot of dysregulation of DNA double strand break repair activity happening, with increases happening or decreases depending on the pathway. These 3 specific microRNAs that we look at is highlighted here in this little hairball network here. And we see that these 3 microRNAs, when you map them to all the potential targets for gene targets for DNA double strand break repair, this is what this shows here. So they have a lot of influence, these 3 microRNAs, on DNA double strand break repair genes. This is what this is showing here. So when we quantified it after an hour and a half, because that's the peak, you might see some peak activity with DNA double strand break activity. So you quantify it by staining with 53BP1, which is the DNA repair protein, and also the genes involved in that. And when you stain it, you can see these foci that might pop up whenever there's a DNA double strand break, because that's where the repair activity is happening. And what we see is that here's the 0 gray, which you don't expect much activity happening here. This is the 0.5 gray, which you see a lot of foci in the nuclei that might happen. And then this is with integumentary treatment and irradiated at the same time point. And you see a significant reduction in DNA repair activity, which could mean a couple of things. One, we physically protected the DNA double strand breaks, which I don't think might not be possible. We inhibited the DNA double strand break, which again, from all the beneficiary things, I don't think is possible. Or we sped up the repair activity, which I'll show you. I think that's the option 3, I think is what happened. So now what we did was, to look at further in the biology, is take these 3D tissues after irradiated, after the treatment, with and without the treatment, and then did some sequencing on them. So you got the mature and the angiogenic model. And overall, what I want you to focus on here is this plot here, and I'll go to the left plot also, but this is the PCA plot, looking at the clustering of whether the 0 gray, so this is 0 gray is the blue, sham, no treatment. The green is the 0.5 gray without a treatment, and the yellow or orange is the 0.5 gray with the treatment. And this is PCA plot looking at the clustering of our overall genes. And what's really nice to see is that the overall globally, with the treatment of the integumentary after radiation, is pushing it closer to the 0 gray level, which is now showing some rescues. In my opinion, it shows that it's trying to push the overall genetic or the transcriptome back to the 0 gray level, which is really nice to see. Of course, it doesn't fully do it, but it's getting closer. So this network plot here is also interesting analysis done with our collaborators, where they took the genes that were significantly regulated when you compare 0.5 gray to 0 gray without treatment. And then they said, okay, what gene from those genes, what genes are not significant anymore when you give the treatment, comparing 0.5 gray to with the integumentary treatment to 0 gray. So basically it means with the treatment, it pushed it back down to the 0 gray level, the genes, and now the treatment basically restored those genes back to the regular level without any radiation. So in a sense, it rescued the damage caused by those. And then what they did once they found those genes is found the overall gene targets for these for the microRNAs, and this is what it's showing. So this is the genes that are involved there, and here you probably can't read the specific pathway names, but the general pathway names are here, where these genes overall regulate different pathways from apoptosis, cell cycle, cell death, and even a lot of mitochondrial related pathways are related with these genes. So that was interesting to see. That is maybe restoring some of the damage caused and rescuing that. This is some general pathway analysis related to this. And what we see, I mean, overall, you can see a lot of this left column here is related with a 0.5 gray versus 0 gray without any treatment. This middle column is now 0.5 gray with integumentary treatment versus 0 gray. So now you've done treatment. And then this is comparing the different radiations with and without the integumentary treatment. So what we see here right off the bat, which is pretty striking, just similar to that network plot with the genes, you can see, for example, there's a lot of increase with inflammatory factors that you see, with radiation, space radiation, which you expect. But with the treatment, you can see these are not significantly different now with integumentary treatment with radiation compared to 0. So it's rescuing some inflammatory factors that are related with this. There's other things that you can see, for example, hypoxia is increased, which is radiation. You expect that. Oxidative phosphorylations increase, really mitochondrial function, which is expected, but with integumentary treatment, it's pushing them now, inhibiting those. So there's other factors here, but this is some of the more striking features you see. Now, this is for the mature model. The angiogenic model has different biology associated with it, so it's like ramping up. It does have already high increased inflammation because it's developing the angiogenic model. So that we didn't see much change happening here, as you see here, but it does with the treatment even push that further down. And what's interesting is the key pathways like cell cycle is suppressed by radiation, and people know that occurs. Now we've actually pushed, within treatment, we push that back down to the regular levels. There's other developmental factors that like EMT and other things that are being pushed up that are saved also in the angiogenic model. So, doing further analysis here, which for the sake of time, we'll just fly through this, but you can see that, looking at pathways that are not significantly changed with the treatment, but were significantly changed with, the radiation by itself compared to your 0 gray controls. And again, the common themes are here with the related inflammatory related factors with the mature model here, and cell cycle and developmentals for the angiogenic. Now here's a specific mitochondrial impact. So looking at the mito pathways for mitochondria and mapping it onto the mature model and seeing how this changes. Again, left column is no treatment, 0 gray compared to radiate versus 0 gray, and this is the integumentary treatment with that. And you can see, for example, the red font pathways are the overall, all the genes involved in metabolism, not the sub pathways, say MOXPHOS, all the genes related. As we showed earlier, like all the genes with OXPHOS shows an upregulation with, radiation, same metabolic activities is reduced significantly, and so on. But, with the treatment is pushing these things back down or suppressing them sometimes. So this is something we're diving into and interpreting now to figure out. Overall, we think this is a good thing because this maybe restoring some of this activity that's happening, but maybe some of this stuff might be too much suppressed. So maybe our dose we gave was a little too high. So we have to figure out the right dosage, which is the next steps. Looking at all the different dosing curves we could do and figure out what's the balance. And of course, interpretation of this is fully in development, and anyone here wants to join and help us interpret all this data would be great. So now, okay, this is all human in vitro data. Does this work with if you go into organisms? I mean, unfortunately, we can't stick it in a human yet, so we go into mice as the next best thing. So again, this is simulated experiments we do for mice that had a normal loading or hind limb. Remember hind limb loading I showed you earlier was the simulated microgravity setup. And then we suspended them like that for 2 weeks. And 2 weeks is a minimum time to allow the microgravity simulated conditions and the muscle wasting to settle in. And then we irradiated them with and without the setup, the microgravity simulated or not. And then we sacrificed the other mice in 24 hours later. So, and we did different radiation types. What's here is now the weights, and the two weights that you probably should focus on is these two were with and without the treatment. So you can see this is the heart. I'm just going to come into play for a second. So the light color is the mice not irradiated, sham, no treatment. This is just hind limb, because these are just the hind limb setup mice by themselves. And this is the sham, no radiation, and this is the hind limb. So we're restoring sort of these, you can see, back to the normal levels higher up. So the weights are kind of be rescued in the heart, same with the radiation we see. One thing is interesting, this is ongoing work with collaborator Jonathan Sisler in his lab at UNC. But he started, and he has some more data, which we're not showing here. But what's really interesting is we see that the DNA copy, mitochondrial copy numbers with radiation, no treatment now, is heavily suppressed in the cerebellum. So he looked at the cerebellum from these mice. And then he said, okay, let me first start really simply by looking at the DNA copy numbers, mitochondrial copy numbers. And you can see this is the 0 gray, no radiation. This is the GCR, and this is the solar particle simulated beam, which is what's the solar flare, as I mentioned, like high energy protons from a solar flare that the astronauts will be exposed to. So you can see that the mitochondrial DNA copy numbers in the cerebellum has been significantly reduced, after 24 hours after this simulated space radiation by itself. Now, what's interesting is when you do the hind limb by itself without any radiation, the microgravity simulation, the DNA mitochondrial copy numbers has increased significantly. So there's a little interplay there where microgravity is increasing the DNA copy numbers in the cerebellum while radiation is suppressing that. Now, when you do the hind limb unloading by itself, which is this one, depending on the radiation, so this, if you just look at the blue color, is the control, and I'll get into the integumentary treatment in a second. But we see that the GCR still is reduced, whether you have hind limb or not, but it's not as significant because maybe you have this interplay here where microgravity is increasing your DNA mitochondrial copy numbers while this is reducing it. And SPE kind of, with the combination of the microgravity, is bringing it closer to your control levels. Again, the interplay is, you can see it maybe here. Interestingly, now without the hind limb setup with integumentary treatment, we see that this is the GCR. We're actually restoring it closer back to the 0 gray levels. Hind limb setup didn't have as much impact. Again, we tried one dose. The dosing might be different, and also maybe some other. Remember, I said these microRNAs associated microvascular effect and cardiovascular effect, not necessarily CNS issues. So there might, for my microRNA signature of 10 to 13 microRNAs, there might be a different components to add to this to actually figure out the best CNS behavior. Now looking at the heart specifically with our collaborator in Austria, Peter Jurek, in his lab. So he took the hearts, and now looking at specifically, we're analyzing this paper should be submitted again at the end of this month. But he first started looking at fibrotic related genes, and then also mitochondrial related genes. So interesting enough, when you look at TGF-beta, which is also related to mitochondrial, not just fibrosis, but you see that the blue here is the normal loaded mice with galactic cosmic rays. This is the integumentary treatment. This is the sham mice, no radiation, sham with the integumentary treatment. You see, we completely rescued or restored TGF back to the normal levels with this treatment. SMAD is also, and then this is the paper showing how these microRNAs are related with TGF-beta signaling too. SMAD has an interesting thing, it's really fibrosis, and indeed it's bringing it, seems to be larger variation, but some interesting interplay here, which we're diving into. MCU is a mitochondrial calcium type involved with mitochondrial calcium activity in the heart, and we're adding more data points here as we're looking into it. But what's interesting is that you see that there's heavy suppression of or heavy increase of MCU activity, which is bad for the heart to have, and when you just irradiate them. This is the integumentary treatment. This is your control, no radiation. It brings it back down. MICU1 is again related with mitochondrial calcium uptake and myocardial ischemia, perfusion injury in the heart, as later shown. And the suppression of this is bad in the heart. And indeed, this is the radiation does this. This is your control. This is the integumentary treatment. So we have more data in the paper that we're putting in there and looking at how this integumentary treatment restores mitochondrial function in the heart, which is really exciting to see. And again, the doses we gave will still be an interplay. And we have other working on the kidney and other organs showing similar behavior with mitochondrial restoration of these microRNAs. So overall, the summary of the work is that, mitochondrial dysfunction in space is a true thing happening. It's pretty universal, depending on the organs. Depending on the organ, you might have a suppression or increase or increase in mitochondrial dysfunction, which either way is bad because of the whole overall dysregulation we see in the body and compensation for certain, things that are occurring in different tissues. And then, the microRNAs might be an avenue where, one, you could use that as a biomarker to show that increased acceleration in mitochondrial dysfunction that we see occur in space, what's specifically occurring for which organs. And then potentially maybe it could be a therapeutic or countermeasure to use to potentially rescue that damage. So maybe an astronaut could take a pill or a shot and help him or her to actually reduce the mitochondrial damage that people experience in space or dysfunction. And then this obviously can be easily applied to therapeutics in the clinic too. MicroRNA acknowledgement, of course, this is done with lots of people, so lots of great people, and the funding from NASA and Trish. And one quick thing I want to bring up, so keep in mind for future exploration. So right now, this is a thing called twin orbit. So I mentioned Scott Kelly and Mark Kelly. And so one thing we're doing is with these group of people, we formed a group called Twin Orbit, where the goal is the person initially, Brent West, Commander Brent West from the U.S. Navy, had approached Chris Mason, since Chris Mason was in charge of the original the twin project, Scott Kelly, Mark Kelly, PI there, where he's a twin, his brother Brad, and he wanted to actually see if we want to do a new twin study. So this evolved now to the team we have here, a bunch of nice people in our science team, which some of you might recognize, some of the people involved, like Doug Wallace. And the goal is now to do a future mission and missions of twins, not just have one twin, does not just send Brent up to space, but Commander Brent West, but also send multiple twins, which we're in the process of recruiting and also acquiring funds to do this. And some of the goals for this project is, to be not only looking at, not only just to send them to space and look at that, but now we can have basically the 1,000 data points where now we look at things related with health, that astronauts we should point out. One of the focuses is mitochondrial, since we know, and aging, since we know mitochondrial disease and accelerated aging occurs, is to now, when we do the missions happening, we've already established this as a for-profit, now established as a nonprofit. But also, by 2025, we're in talks with a few folks to be like, all right, then we could send 12 twins into space. And then now we got identical twins, and we imagine about in planning all the mitochondrial related activity we can look at and functions we can look at, and eventually do more missions and relate to this. So this is something we're in fundraising mode, so people know people who might want to invest. That could be something we can talk about later. But that's it. So thank you for your attention, and I might have went over a little bit, but hopefully not too much. I'll stop.